I'm Dr. H.K. Tyagi, principal of a college. I'm today talking C3 between C3 and C4 plants. Between C3 and C4 plants, fixation of CO2 takes place only by only two ways. First, in C3 plants, it takes place by ribulose CO2 acceptable, and it is accepted by ribulose CO2 acceptor, and in the last, does not contain Cron's anatomy. But in C4 plants, there is no ribulodiphosphate. First of all, it takes place by mesophyll cells. By mesophyll cells, oxaloacetic acid is formed, and mesophyll cells form phosphoenol pyruvate. In C4 plants, Cron's anatomy is present.
Hello. Start हो जाए.
Dear students, I am talking about chloroplast. Chloroplast and peroxisome and mitochondria takes place three organelles in the C4 in the photorespiration. In photorespiration, chloroplast, peroxisome and mitochondria takes place. First of all, in chloroplast CO2 is accepted and it takes place into Calvin cycle. Then diphosphoglycic acid is formed. It goes into peroxisome. In peroxisome, it takes place by peroxypyruvate, then glycolic acid. Then glycolic acid oxidase is formed and CO2 and H2O are released. Then serine is formed. It goes into mitochondria, and serine goes in mitochondria, and in mitochondria, first of all, 2 glycine are taken, and CO2 plus NH3 they are released. Respiration topic. Respiration is a very important topic, and it takes place in examination. First aerobic respiration, then anaerobic respiration. In aerobic respiration, 6O2 in the presence of enzyme they form H2O and 673 calorie energy. In this respiration, first is EMP pathway. EMP pathway means Embden-Meyer-Parnas pathway. First of all, phosphate isomerase is formed, and ATP is changed into ADP. Then glucose is formed, and from the glucose phosphate there is formed phosphoglycomutase. In phosphoglycomutase, ATP is changed into ADP, and fructose 6-phosphate is changed into 1,6-diphosphate. And in this method, aldolase takes place, and ATP is changed into ADP. In the last, phosphopyruvic acid is formed. Phosphopyruvic acid 2 molecules. वो दिखाओ लाइट. Or TCA cycle. First of all, NADPH formed NAD in the presence of Mg, and phosphoglycic acid kinase enzyme is formed. Then hexokinase enzyme takes place, and 1,3-diphosphoglycic acid is formed. Then phosphoglycomutase is formed, and phosphoglycic acid kinase is formed. NAD is changed into phosphoglycic acid is formed, NAD is changed into NADPH, and enolase is formed. Pyruvic acid, and in the last, phosphoric acid and pyruvic acid is formed, and in the presence of ADP, ATP is formed, and 2-phosphoglycerol mutase is formed, and in the last, pyruvic acid is formed.